All right, guys, I made you a photosynthesis cartoon and I'm going to go ahead and talk you all the way through it. All right, so here we are. We're in the chloroplast. This is the stroma of the chloroplast. This is a thylakoid. We have a cross section of it here. Remember, the thylakoids are those little stacks of things inside the chloroplast. A whole big stack of them together is called a granum. They have a membrane. This is a normal phospholipid bilayer membrane. It is hydrophobic on the inside and hydrophilic head groups. Remember, because it's hydrophobic on the inside, no matter how small a molecule is, if it's charged, it's not going to be able to cross that membrane because of this hydrophobic area. Small gases and water can cross the membrane. Up here we have PS2 with these little pigment molecules, the green things, and a reaction center. Over here we have PS1 with these pigment molecules and the reaction center. Down here in the membrane, we have the ATP synthase enzyme. So now we're going to talk through what happens. So there is water in the thylakoid lumen. Remember, the lumen is just the inside. That water is split. It's split into hydrogens and oxygen. Each hydrogen is really a hydrogen ion and an electron. Those hydrogen ions contribute to the hydrogen ion gradient that we're going to make, and so they sit here in the inside of the thylakoid lumen. The oxygen is 1/2 of O2, so another water will be split and it will make an O2 molecule, and remember, gases can leave. The other thing we have from the hydrogens is the electrons, so we split it into a hydrogen ion and an electron. These electrons are loaded onto the reaction center of PS2, and I have them here as cats because I like cats. The next thing that happens is sunlight is shining on the plant. These accessory pigment molecules, or these antenna pigment molecules, can absorb a photon of light energy, and they pass it like a hot potato from pigment molecule to pigment molecule, where it ends up at the reaction center. When it ends up at the reaction center, it causes these electrons to jump up in energy state, as you can see here by the supercats with the stars. When these electrons jump up in energy state, two different things can happen. First, they can fall back down to ground state and give off heat and fluorescence, or they can go to a primary electron acceptor. In this case, they go to a primary electron acceptor, being the electron transport chain. So they move onto the electron transport chain. Again, these are high energy electrons. They've captured the energy in that photon from that photon. So what happens is they move down the electron transport chain in a series of redox reactions. Remember, when something gets an electron, it is reduced, and whatever is losing the electron is oxidized. Every time it moves down the electron transport chain, it loses a little bit of that energy. That energy is used to drive hydrogen ions from outside in the stroma into the inside of the thylakoid lumen. Again, it loses a little bit of energy. Then it moves again to the next electron carrier. Another hydrogen ion is driven in, utilizing the energy that is lost by those high energy electrons. So now we have these electrons here. All of the energy that they captured at PS2 has been used to drive these hydrogen ions in through the membrane into the lumen of the thylakoid. We have our electrons now sitting here at the reaction center of PS1. But again, this is a photosystem. So light energy in the form of a photon is absorbed. That energy is passed from pigment molecule to pigment molecule, ending up at the reaction center, which causes these electrons to jump up in energy state. Remembering, the options are they can fall back down to ground state and give off heat and fluorescence, or be picked up by a primary electron acceptor. In this case, they are picked up by NADP+. The enzyme that does this is called NADP+ reductase. So it reduces NADP+. NADP+ grabs the two electrons and a hydrogen ion to make NADPH. So now we've made NADPH, and we have a whole bunch of hydrogen ions inside of here. This keeps going and keeps going because water keeps being split and electrons keep being loaded here and moving down the electron transport chain. Now let's talk about what's going on with the ATP synthase enzyme. So we have this large hydrogen ion gradient where there's a lot of hydrogen ions in the inside of the thylakoid lumen and barely any outside. Remember, they're being driven in by the electrons moving down the electron transport chain, and they're being removed from the outside by that NADP+ reductase, making NADPH. So the hydrogen ions would like to go from a high to a low concentration. However, they cannot cross this membrane because they are charged and they can't get through this hydrophobic space. They can, however, travel through this ATP synthase enzyme. When they travel through the ATP synthase enzyme, they are flying through there. What happens is they cause a mechanical turning of the bottom of this ATP synthase enzyme. That mechanical turning, the energy that is created there, is used to join an ADP with an inorganic phosphate, making ATP. So now we have created NADPH and ATP. Let's go on to our next step, which is the Calvin cycle. So in the Calvin cycle, we created this NADPH and the ATP from our light reactions. And we have this 5-carbon molecule that is sitting and waiting. This is a cycle. So we start with something, we turn all the way around, and we end up with that same product. This is called RUBP, ribulose bisphosphate, a 5-carbon sugar. In the plant, CO2 comes in, and the enzyme Rubisco can attach this carbon to this ribulose bisphosphate, making a 6-carbon molecule. The oxygen leaves the photosynthetic organism. This 6-carbon molecule is not very stable, so it immediately breaks in half into 2 3-carbon molecules called PGA, or phosphoglyceraldehyde. Now, remember the whole point of what we're doing is photosynthesis, which is making carbohydrates, mainly glucose. And we just made all these high energy molecules. What we want to do is take those high energy electrons from the NADPH that have that energy from those photons of light energy, and then also the energy on the bonds in ATP, and add it to these molecules. So what we do is we add 1 ATP, the energy from 1 ATP, and the high energy electrons from 1 NADPH to each one of these PGAs. And we end up with a different molecule called G3P, or glyceraldehyde 3-phosphate. So although this is 3 carbons and this is 3 carbons, they are different molecules. Remember, shape is highly related to function. So basically, you can think of G3P as a PGA with a whole bunch of energy on it. Some of those G3Ps leave and go make glucose and other carbohydrates. And usually I like to talk about glucose because that's the easiest one. So we're going to say we pull out 6 carbons because glucose is a 6-carbon molecule. The rest of the G3Ps are cycled around to remake RUBP. And that takes energy from ATP. So good thing we made all these ATPs and all these NADPHs in the light reactions. So now I want to get a little bit more detailed and talk about the numbers of these things. So since we're pulling out 6 carbons here to make a glucose, and this is a cycle, that means we're going to have 6 carbons come in. So we have 6 RUBPs waiting, and the plant or photosynthetic organism brings in 6 CO2s. That leads to 6 unstable 6-carbon molecules, which all immediately break in half, and we end up with 12 phosphoglyceraldehydes. Each one of these gets energy from ATP and NADPH. So we use 12 ATPs and 12 NADPHs, and we end up with 12 glyceraldehyde 3-phosphates. Now again, we said we're going to pull out 6. So 2 of these leave, and we're left with 10 G3Ps. So we have 10 3-carbon molecules. Our total is 30 carbons. We use the energy from 6 ATPs to reorganize these into 6 of these. This is 6 5-carbon molecules. Again, 30 carbons. So we had our light reactions where we created our ATP and our NADPH, which were our energy that we put onto the carbons from this CO2 to make glucose. I hope this makes it more clear. Happy studying.